All right, we're going to go over real quickly the procedure and kind of background for the Peroxidase Enzyme Lab so people know what's happening here. So again, this gives us our overview in terms of what we're looking at. Make sure you get the correct linking code when you go into it. So looking at what we're dealing with here is the idea that we have cells, that all animals have cells, and that cells need energy. So they get their energy from the form of this molecule called glucose. So it goes into the cell, we break it down. One of the things that happens though is that in the process of breaking this down and releasing the energy, we also produce a molecule called hydrogen peroxide. And hydrogen peroxide, H2O2, it turns out is bad. It's actually toxic for our cells. So we don't want to have hydrogen peroxide in our cells. So we're going to attempt to get rid of that. So the way we get rid of that is we make this enzyme called peroxidase, ASE. Remember, ASE is enzymes. It acts as an enzyme to catalyze the breakdown of hydrogen peroxide. So here's just kind of an example, some visuals you can see with the hydrogen, the horseradish peroxidase there. So we're going to take, in this case, they take turnips or something like that, we take the hydrogen peroxidase plus, pardon me, the peroxidase plus the hydrogen peroxide together, that breaks this down and it turns it into water plus oxygen. The oxygen goes away, the water's still kind of there. Right? So we can look at water and we can say, yeah, that looks like water. So if we wanted to do this reaction though, one thing that's important to understand is that hydrogen peroxide looks pretty much the same. So there's no visual way to tell the two apart, H2O2 and H2O. They look pretty much identical. So how do we know a reaction has actually happened here? Well, we need to add something that changes color, and then we want to measure that change of color. So we can do that using an instrument called a spectrophotometer. And here's a picture of a spectrophotometer. So we need to explain a little bit how spectrophotometers work, how they enable us to measure that color change. So we use a molecule called guaiacol. So guaiacol normally is clear. And so you take that guaiacol, and if you combine it with oxygen, remember we got oxygen from the breakdown of hydrogen peroxide, then it turns color. So we can get the oxygen from the hydrogen peroxide breakdown plus glycol, and that lets us know that that has happened. So inside the spectrophotometer, what we see happening is we've got a light source and we've got a detector. And so what we're measuring is we're measuring the percent of light that is transmitted. So what that means is if you shine a light through there, it's going to tell you get so much light coming through. Some of that light won't go through and it'll be absorbed, and anything that's not absorbed, pardon me, anything that's not transmitted has been absorbed. So we can take the clear solution out and we can put in, our solution that's got guaiacol in it, and we can measure it and we can see, oh, the percent light transmitted, right, has gone down, which means that the amount of light absorbed has gone up. So as this goes down, the amount of light absorbed goes up. It relates to the amount of light that is being absorbed first. That means less light is coming through. Okay, so knowing that, we can then, in addition to our hydrogen peroxide and our peroxidase enzyme, we can add this guaiacol. So it's colorless. Reduced means it doesn't have oxygen attached to it. And then we throw it in there. We get that oxygen and water coming out. The oxygen combines with the guaiacol. It's now oxidized, and it turns kind of this amber color. So what that means as far as our experiment is that if the enzyme peroxidase breaks down hydrogen peroxide into water and oxygen, and the oxygen combines with glycol, causing it to change color, then the more enzyme that's present, the faster the color change will happen. This results in greater light absorption, right? So we get less light going through. Okay. So what we're going to do is you're going to navigate to the website. You need to, I don't know which web browser works best, but you need to register, create a student account, and then follow one of the appropriate linking code to match with your last name. If you have questions, let me know. And that gives you the background on the lab. Thank you.